Time for part 3 of the Comparative Genomics lecture for graduate genetics. So I'm going to give you some examples and just talk about a few more odds and ends. The only new thing is a little bit about hidden Markov models and protein domains. And so in particular for using comparative genomics, I mentioned that for studying viruses, but also for studying bacteria. Even for really well-known bacteria like E. coli, many of the proteins, like I think a majority of the genes and proteins that they have, we don't really understand what they do. I mean, even with human and mouse, it's true that we have ideas about most genes now, but we don't really know what all the processes that they're involved in, let alone for really diverse species. And we discover new species of bacteria and viruses all the time. So we can use comparative genomics to try to figure out and predict what does a gene do, what does a network of genes do, or what does a part of a gene do. And so one approach for this, and this is something that we actually do with the Gordon lab, is to try to use hidden Markov models to look for protein domains within a gene. And that's an advantage because although from really diverse, viruses and bacteria and humans, the primary nucleotide sequence might be completely different. Like there's no way you could ever align them. They're completely divergent now. But the domains that's built, the protein domain, is actually similar. So domains that our proteins have are the same as bacteria and the same as even viruses, even though their sequences are completely different. So there's these conserved protein domains, or chunks of sequences that have a conserved function, and we'd like to be able to track those. And the way that you can do that, and you guys have probably used these tools as well, is to use tools like PFAM, which stores what's called a profile HMM, or model. And then you can run your sequence through and ask, like, oh, what protein domains are present in my sequence against some reference set. So anyway, this crazy clustering graph is showing for a ton of different, this happens to be, I think, viruses. And here's a lot of different identified domains within that. You can decompose this matrix and find clusters both of which viruses are similar and cluster together based on their domain architecture. Or you can flip it around and ask the other question, which is which domains are similar based on what viruses express them. And so you can use this to answer a lot of different questions. The advantage of domains over genes, again, is that genes, you might not know what the orthologous gene is in two different or many different diverse organisms, right? And if you can't say that they're the same gene, you can't do this analysis, right? All of these analyses rely on you being able to align something in a single column or place a number in a single column. But if you don't know that two organisms actually do have the same gene, you can't do that. But the great thing about domains is that because of the way hidden Markov models work, you can find out whether that domain is present or not, or where it is and anything else about it. Because hidden Markov models let you look for very fuzzy things, if you will, not specific sequences, but rules that are a lot harder to visualize. So in that vein, I want you to know a little bit about hidden Markov models. Here, specifically, I use them to look for protein domains, and you guys can use a program called HMMER, either just online, or you can run this on a cluster and to search for domains. One of the people that's really done a lot with protein domains is Sean Eady. He used to be at WashU. He's now at HHMI, so a very important figure in this. And this is from a paper that he put out just to really get people thinking about hidden Markov models and how powerful they are. This is, of course, a Bayesian approach because it's, you know something and then you're trying to figure something out. So you can see the hidden Markov model up here. There's a bunch of different probabilities here. And what this is trying to illustrate is, this isn't like a real, this is just like a kind of a fake hidden Markov model just to illustrate the point of how they work. So this is a hidden Markov model to try to detect the end of an exon and the start of an intron, therefore the 5' splice site. So 5 here indicates the location of the 5' splice site. And then E means this, we think this base is part of an exon, and I means this base is part of an intron. So there's a lot of funny stuff happening here, but it's actually, once you get the hang of it, it's pretty easy. So these numbers up here are called emission probabilities, and that just means an exon, within an exon, there's a completely equal chance for there to be an A, a C, a T, or a G. Let's say, again, this is kind of a fake hidden Markov model, but it's close to what you could really use. And so you're saying within an exon, like any single base within an exon, the probability of being A, C, G, or T is the same. And in this case, within an intron, he's saying that C's and G's are less frequent and A's and T's are more common. And then for the 5' splice site, we know that G is really, really common. And ever so often, 5% of the time, you might have an A, but there's never a C or a T. So, okay, that's one bit of information that we know. And we can get these numbers, we can get these emission probabilities just by looking at annotated sequences, right? So if we look at, let's say, 100 different 5' splice sites and say, okay, let's look at the sequence before it and the sequence after it, and let's just get these, let's just count these things up, and that gives us our probability. So it's relatively easy to make this top part of the diagram. Okay, there's another part of the diagram as well, and that's called the transition probabilities. So what's the transition, if you're in an exon, what's the transition of staying in an exon or going to the 5' splice site, which is your only options, right? If it was something more complicated, you could go somewhere else, but here it's just like, oh, we're going to stay in the exon, which is, the chances is really good. In this model, it's 90%, and the chance that you go to a different one is 1%. And again, you could figure that out just by looking at many different examples and calculating the probability in those examples, the frequencies. Okay, once you've gone into the 5' splice site, which is just a single base, the transition probability is 100% that the next one is going to be the intron, right? And just that's by definition. And then once you're in an intron, there's 90% chance. In reality, this probably should be higher, right? Because introns are usually quite long. But it's saying that in this model, there's a 90% chance that you're going to stay in an intron and only a 10% chance that you're going to leave the intron. And in this case, that's the end of the model. So that's it. So you've got states, E, 5, and I, that happen in a sequence. And you've got transition probabilities and emission probabilities. So here we go. We can input a sequence into this model, and then there's special algorithms that do this for you. But basically, one way you can think of it is just for this particular model, you can basically have the 5' splice site anywhere from here, this T, all the way to this C, right? Or all the possibilities. Now, many of those don't work at all because they're on a C or a T. So if we just take the G's, which is the blue lines here, it could either be 1, 2, 3, 4, 5, or 6. And so what you can do now is, now that you have all this data, you can say, okay, what is the probability that, let's say, if we put this sequence into the model, what's the chance that this is the correct location of the 5' splice site? What's the chance if this is the correct location? What's the chance if this is the correct location? So we do that for every possible site. It's called the posterior decoding. And you get a probability or a percent. And then you just say, oh, well, might as well choose the most likely one. So the maximum likelihood is right here, is the lowest p-value, and the highest posterior decoding. It's this location right here is the most likely, fit for this model, where this is the exon, this is the intron, and this is the 5' splice site. So you guys might be able to use this to build your own hidden Markov model. It doesn't have to be one that someone's already built. You can build your own one pretty easily and evaluate it in R, and evaluate it in MATLAB, and evaluate it in your own Python or C#, whatever you like, and use it to discover cool things. So whenever you have different states that are transitioning, and emission probabilities, you can build a hidden Markov model like this. So you can apply it in lots of different places. So it's another tool that you should have in your arsenal. And in the meantime, use PFAM and HMMR to look for domains and sequences that you might be interested in. Okay, so I showed you a second ago that this is one thing that we do. And I'll just show you one more figure for this, which is that here's all of the reference viruses, which are all very small or relatively small. And it's showing each row of this funny looking figure is showing one virus. And so there's about 1,600 viruses in this picture. And every color is indicating a type of a viral domain that's been identified with hidden Markov models. And then they've been clustered in such a way so that similar ones fall close together. And so if I zoom in a little bit more, you can see lots of individuals. So these areas that are highly conserved, you're kind of looking at domain synteny here. So this is a particular domain, this is another domain. And it's saying that like these 50 viruses all have exactly the same domain structure. I think these are coronaviruses down here. These are like mycoplasma and things like that. But you can actually see what their domain structure is, what domains are common, what domains are different, and how different viruses adjust. And here's an actual look at, I think it's zoomed into this region. And you can see some of these details of like, oh, it's the bacula domain or the FTSJ domain. You can see all the details of that. And some have repeated domains inside the virus. But again, you can do this for mammals, you can do it for bacteria. It's another way to try to find similarities and differences. And very easily, if there's an unknown domain that falls into one of these graphs, you might be able to say, oh, you know what, this really is similar to this other domain based on its neighborhood and other factors. So another tool you guys can use. Okay, so shifting gears, we'll go from bacteria now to primates. There was a lot of, after the human genome got sequenced, there was a lot of interest to sequence primate genomes. Of course, this was insanely expensive at the time, so there was a lot of pushback for it. But eventually, curiosity killed the cat, right? People were very interested in how are we different than these wonderful creatures who, in many ways, act very similar to us as this little guy's doing. I know this is what you guys are doing right now as you listen. And so the argument was made that, if we understand the differences between these genomes, we can potentially make a big stride against important things. So, for example, if a human gets HIV, we end up getting AIDS, whereas if a chimp gets HIV, they're very unlikely to get AIDS, right? It doesn't really hurt them. Same with influenza and hepatitis, plasmodium, all those things we get. We're pretty susceptible to infections and we have a strong response, whereas for chimpanzees, they have very little response or just resistant. And so what in their genomes is causing this? You can see a few other things here. This one I was very surprised to see. So menopause, of course, all human females get menopause, but in chimps, it's very rare. Gastroenteritis, Alzheimer's pathology is incomplete there. It's unclear if they live, are they reaching the same age? Epithelial cancers are pretty rare. The last few I'm not so sure about. It could be an age-related thing. But certainly for disease, things like infection, it would be interesting to answer those questions. So, but now that you guys know about multi-species alignments are tricky between closely related species, know that this is going to perhaps not tell us so much because, even amongst mammals, there's a huge amount of conservation, let alone between humans and primates, which are really closely related. So what regions of the genome do change really fast? And these are some of the places we can really look for massive changes. And these are kind of the big ones for humans. So near the centromere, there's regions there. You guys might know that in the reference genome, it's very hard to sequence near the centromere, and that's because there's a lot of duplications. So as you get close to the center of each chromosome, the pericentric regions, it's very hard to look there, and the duplications are thought to change pretty rapidly. So the very center. Another place that's very hard to look at is on the very ends. So near the telomeres, so just inside of the telomeres, there's these subtelomeric repeats, and there are other regions there that just the mutation rate is very high, and it's very tricky to sequence, and it's also thought to change very rapidly. And then the last place is kind of interesting, is the Y chromosome. If we were here in person, I'd ask you, do you guys know what is changing about the Y chromosome, right? So this is obviously the male determining chromosome. The thing that's changing about it is that it's getting smaller. So men, being defined as male, you basically just need a Y chromosome that has SRY, a gene that says like, okay, you should be male. Because everything else, both sexes need. And so more and more genes are getting moved off of the Y chromosome into other chromosomes. So the Y chromosome is getting smaller and smaller. Oh, and I should have drawn this picture for you right away, but just a quick illustration of where the subtelomere and pericentromeric regions are. So these are some of the places where you can look for massive changes, but otherwise, there's not a whole lot of change. We're pretty similar. Okay, so along the bottom here, if the hypothesis, there's basically 4 hypotheses for why we are different than primates, let's say chimps or apes, genetically, right? And so in addition to changes in these regions, which we don't really understand and are hard to look at even now, you can imagine that humans are different than apes because of a gene loss, so something that they had and is mutated in, is lost in us. A gene gain, like a new gene that we have that chimps don't have. Mutations in genes that we both have, or differentially regulated genes. And the answer to this question, of course, is that the answer is all of them. We have examples of all these. And sorry, I should have said that the big question here is what gives us our humanness? What makes us human, and what makes apes apes? So I'll show you a few examples of each of these. They're interesting case studies. I don't think there is a smoking gun. I don't think there is a single, and I don't think most scientists think that there's a single gene that gives us our humanness. But there are that seem to be more important. One example is this sialic acid hydroxylase. It's involved in sialic acid biology. And so we have a big frameshift mutation that basically deletes this gene. So we don't have this particular gene, but chimps and gorillas and most other mammals do. And this is called new 5GC. This is the product of this gene. And apes have a ton of it, but we have almost none. It seems to generally be localized on the surface of epithelial cells. And interestingly, there's some data that it looks like it constrains brain size. So possibly that we bleed this gene and we have less of this sialic acid could be leading to our larger brains. It's a hypothesis. Okay, could we be, gene loss is relatively easy. It's easier to find, right? A gene that we both have, but ours is broken, ours turned into a pseudogene, basically. Gene gain's a little trickier because where would it come from? But one place it could come from is by a duplication. And so this is a really interesting gene family. We basically have way more copies. So some lower order primates have just one of this gene. And in fact, all mammals just have like one of this gene. We don't know anything else about it. It doesn't really have any homologs in any other mammals that we understand. So there's just like one gene in other mammals. But then between monkeys and us, it got duplicated many times. So there's like a 20 kb segment on chromosome 16 that has most of the duplications, but then there's a few that are scattered in other places. When you do the selection test that I told you about earlier in part 1, the KAKS test, so this is looking at non-synonymous rate of mutations over synonymous rate of mutations, the number is really positive, meaning that this gene is under really high positive selection, meaning there's a lot more mutations here than you'd expect by chance, or compared to a random model. So this is called the Morpheus family. Morpheus, because of course the matrix. No, I actually have no idea why it's called the Morpheus family. And you can see that, it has something to do with nuclear pore biology. We don't quite understand what, but it's expressed in most of our cells, and you can see these our very close relatives have more of it. It's spread across a few different places, whereas old world monkeys that are less related have much less of this particular gene or gene family. So maybe very likely this is one of the causes for our humanness. Well, what if we both have the same gene, but we have different mutations? Obviously, that's the case for many of our genes, almost all of them. One way to look for this, I'll show you a quick genetic thing, is called selective sweeps. So here's your population, right? So this is a particular gene, let's say, or a region that has several genes in it. And each of these colors is a different allele. And of course, on one particular chromosome, you have a set of a few different alleles that are all linked together on the same chromosome. But let's say this particular arrangement, in particular this allele that I've starred, which will just make a pink star, perhaps next to this green allele, this really increases the fitness. So what you can imagine is that you get what's called a selective sweep, where now this is really strongly negatively selected for, so you get purifying selection. And you see what you've done is in this region, because all this is linked, you've lost all the other diversity in this, at least in this region. As you get further away from the site, of course, you start to see more and more diversity. But so over time, as mutations start to reoccur, you see this region that's, like, of course, very strongly negatively selected. And then even the region around it is also, there's this local reduction in diversity. So that's what a selective sweep kind of looks like. So that tells you that some part of something going on here was, really selected for, and it's kind of swept all the diversity out of this region. So this is what you would see. And so there's been many attempts to kind of look for all the selective sweeps compared to us and different kinds of primates, and to assume that, at least to look into the hypothesis that those regions could be mutations that are giving us our humanness. One of the most popular of those is FOXP2, the transcription factor. And this is really cool because this is probably one of the better hypotheses. It's certainly one of the more compelling ones. Whether it's actually true or not, it's hard to know because we can't really do an evolution experiment on a primate yet. I don't even know how you would do that exactly. But what we do know is that if you take a human and you mutate this gene, this is a gene that looks to be a selective sweep event has occurred, or a mutation compared to monkey. So in this particular family, individuals that have mutations in this gene have a really bad speech impairment. They don't actually have other mental illness, but their speech is very much impaired. And so you can just see kind of a nice pedigree here. And then this is just showing that there's some mutations. There's fairly little mutations, but the humans have these 2 mutations. So one hypothesis, people like to call this is the speech gene. And so mutations that we gain in this genes have allowed us to have speech. This is probably a good time to remind you guys that, again, there's no smoking gun. It's just like there's very few real monogenic diseases. I shouldn't say real. It's very rare for people to have monogenic disease. It's very unlikely that our humanness was caused by one thing, right? Our humanness was caused by many different factors, and these are all examples. But similarly, FOXP2 doesn't just control speech, it probably controls lots of things, but it appears to be the case from this pedigree that if you mess with it, speech gets messed up. And the last thing is regulation, right? You could have a gene that there's really no difference in the gene itself, but it's regulated different, right? You have more expression in one of the organisms, one of the species, and less in the other. So the tricky thing to figure out is how do you express gene regulation? Like how do you find the neutral rate of gene regulation? And there's many algorithms out there that try to do that. I just don't understand them. So, but it is another question you can ask, but you have to understand neutral evolution in terms of regulation. And so that's a little bit of thoughts about using conservation to ask questions between primates and humans. And of course, it's a combination of all these four things, or perhaps other things that we don't yet understand that are leading to our humanness. Okay, the last subpart of part 3 is just a little bit more about comparative genomics within humans. And so obviously, a lot of human genomes have been sequenced. So is there anything general that we can learn? And these are the kind of questions that people have asked in the past. I think this is kind of interesting, but it makes me, it's just not the kind of questions that I personally like to ask in my lab. So it's a little harder for me to connect with them. But certainly I think in the vein of, trying to learn more about our species, I think this is interesting. So here's some of the questions. Are there subclusters of the human genome? Are there a proxy to predict drug responses? I think that's interesting, and certainly precision medicine has attacked that, although my personal thought is that we need to go way down from subclusters down to N of 1 to really predict drug responses. Is there something like a race or geography? Is there a correlate to race or geography within the genome? And can you actually see migration events within the genome? They're all reasonable questions, and as all the human genome sequencing was being done, a lot of papers tried to answer them. So here's examples of some of those papers. And so to get at this, just like I showed you, the human genome versus the mouse genome, or the human genome versus the primate genome, here's kind of a look at human genome versus human genome. So what is the nucleotide diversity? I think you guys already know this, but I'll have you guess in a second. And then what do you think the fraction of that diversity, how much of it is explained by continental populations, right? Like Europe versus Asia. Does that explain the nucleotide diversity? So the nucleotide diversity, this is something that you should probably memorize if you don't know it already, is between you and the person, in your apartment or sitting next to you when you are back in classes, or between you and me, is about 1 in 1,000 base pairs. So every 1,000 base pairs, we're likely to have a variant between us. And that's why over the whole genome, most two people have in the order of magnitude of about 1 million, a few million variants compared to each other. It might be a little bit higher than that. This is probably not putting a whole lot of weight on pericentromeric and subtelomeric regions. But so about 1 in 1,000, so 0.1%. How much of that is explained by continental populations? It turns out fairly little. So most of those 1 in 1,000 base pairs is something other than, you're from Europe or you're from Asia. And you can think of the picture as something like this. So if continental populations are the orange and the blue, yeah, there's a little bit of diversity out here, genomic diversity, right? These variants that are specific to these, but the majority of them are shared. And this is probably just because we have, a certain background rate where we're getting variants all the time, and that it's really early events that led to most of our diversity before we migrated out to different areas. And so our big diverse pool happened early on, perhaps. Now, even though the majority of our diversity or genomic diversity is not based on populations, like continental populations, it doesn't mean that we can't see markers for those populations, right? So there still is a few percent, 5 to 10%, that is explained by continental populations. And you can use those variants to actually map out, and this is kind of cool. This is a principal component analysis among some of those variants that do kind of co-vary with continental origin. And you can actually see this is colored, these are colored the same. So you can actually see England and France and Great Britain, Belgium, Germany. They kind of actually line up in these principal components, much in the way that Europe is actually set out, which is pretty cool. So there is a representation in our genome of this continental diversity, but it does not explain, nowhere near the majority of our nucleotide variation. And this is just another way to show how that looks. Okay, so that is it. So the conclusion for that section is using race or geography is not so effective as examining the individual alleles directly, because there's a lot more information in the actual alleles than just which continent somebody has come from. And that means that trying to use race or geography to predict drug response is actually not the best for most drug responses, right? Because unless you have a gene which happens to have one of these alleles that is very continentally segmented or segregated, it's probably not going to be an effective way to figure out who's going to respond and who's not going to respond. Instead, just going down to the individual variants is going to be much better. Okay, so that's the end of the 3 parts. You might notice in the Prezi under the tree there that there's something interesting that you should take a look at. I hope you guys will email me if you have any questions. And last but not least, I'll leave you with this image. My daughter and I like to play with, my 5-year-old daughter and I like to play with 3D animation stuff. So we did this little review of comparative genomics and the important tools. So remember these things and be well.